Okay, I can see most of the participant is from aquaculture background. So for my job is very difficult to convince, but still I will try to make it more interactive. And I will assure you, EDN is a hot topic in resource management, particularly fish resource management. And well, so today's the rapid changing of the world, the understanding and conserving of the planet biodiversity has never been more critical. And this biodiversity has an integral part of our daily lives. It provides essential ecosystem services, and also millions of people they depends on this. And nowadays, it's a big challenges to monitoring management with effective manners. So that's where this environmental DNA comes into the picture that revolutionize our approach, how to manage this biodiversity, conserving and things. So my name is Mukesh Bhendarkar. I'm currently working with AZTI, Spain, and I'm also affiliated with ICR, Government of India. And I'm happy to be here to discuss about how the environmental DNA transform Indian fisheries management. Well, the topic says Indian fisheries management, but it applicable for all setting, like a tropical countries, whichever is more or less similar to Indian fisheries. Okay. So my presentation would be on the state of Indian fisheries. We can have some eDNA approach, how it will help, the perspective, the advantage and disadvantage, and then finally we will wrap up with the concluding remarks, how eDNA can do it. So let's take a closer look of Indian fisheries. So India is the second largest fish producer in the world, and also is the top inland fisheries production. So it show the data show it's a major place, one of the major player in Indian fisheries and aquaculture as well. And when we are looking about the Indian global fish diversity is contribute the 12% to the global fish diversity. Now also out of 36 biodiversity hotspot, the four are in India. It also have extensive inland and marine resources. It comprises the huge area of the reservoirs, ponds, canals, the huge networks of rivers, and also the floodplain wetlands. When we are looking about the marine resources, the one of the longest coastline and extensive resources support the millions of fishermen in India. And looking at this, that is no pointer. Okay. So we can see clearly how it steadily grows the total production of India from some decades, and it shows is continuously growing. But if you look at the second graph, this production mostly contributed by aquaculture. So it sounds good. I think it's but the marine production is continuously stagnant. So why it is because, well, the in one side is progressing and in second side is stagnant. So there are multiple reasons to staggering this production of marine production. So what are those? So well, it's not in all about the India. Overfishing is the major problem that leads to many species is overstock and also depleted. The second the challenges is pollution from agriculture runoff, industrial waste, and the plastic pollution. There are many things, and of course, the invasive fish is also one of the threats to divorce the endemic species, indigenous species. habitat destruction through barriers, construction of dams, and industrialization. Then these are the list that the major issue that decline the Indian fisheries. And of course, the climate change is now established that pose uh, pressing stress on our ecosystem, and it affects all, well, right from the spawning ground, the feeding ground. It does not affect only that sense, but in larger, most of the species they put out of their native range, distribution change. So these are the, and also extreme events. So these are the major issues that we are suffering. And ultimately, what would be uh, the results is declining per person, per fisherman. So it is declining because the biodiversity is declining, the diverse, because is known Indian fishery is known for diverse fisheries. So how we can manage? So now the monitoring these wide resources using conventional method may not be compete or pace with the current ever changing aquatic ecosystems, because these methods relying on visual observation, the counting method, and it is more like a destructive invasive species, the laborious. We cannot apply in some places, and also destructive. It also difficult to identifying the cryptic species. Well, this method is somehow, some extent effective, but there are many limitations, like we saw. So for this, we need some effective and efficient method to monitoring these resources. What would be? So this is the eDNA, the environmental DNA. Just take a cup of the water from the rivers, ocean, reservoirs, and we can get the wealth of ecological information that would help to conserve this fisheries. So what is eDNA? How we will analyze this? So eDNA is the DNA extracted from the environmental sample without capturing target organisms or even without seeing them. So it just the in general, when species moves around the environment, they lose the genetic traces in the form of DNA, and that DNA shed from the scale, mucus, some activities, and we can have just take that genetic, collect that genetic DNA and find out which species are there. So you can see this picture and you can clearly see. So this is totally non-invasive, cost effective. You can just take a water and get the species. So is it only the taking water? The collecting water is the one thing, but how we will analyze next? So as soon as the collecting water sample, we have to filter them with different filter and then extract the DNA out of from that water. And then based on our study, we have two options. So in one set, if you want to the single species survey, so it's using the specific markers to identify single species. And this method is mostly used for identifying invasive alien species, and it is mostly applied right now. And second one is biodiversity survey, where the small DNA fragment we sequence and get the simultaneously multi-species. And it is mostly used in the ecological, what are the composition and diversity in the available resources. And finally, we will get the bioassessment survey. So these are the methodology, what we call eDNA methodology. And you can see here what are those advantages when we are comparing the conventional method. So, as we know that the India is known for the diverse geographical features, right from highest raining region to desert as well. So, monitoring these resources is difficult. Difficult. So, using eDNA, there are many various promises that we can manage. So, first, the eDNA can establish the baseline survey with each have region, specific region. So we can get the total biodiversity. The regular eDNA monitoring also can detect the changes of species abundance and composition over time. And it's a continued process. It's also effective view detection of the alien species or endangered species without seeing, because the endangered species are rare in abundance in the environment. So we cannot get using capture basis, using net. So eDNA can help us. It also act as an early warning system. So this application can be used in aquaculture also for specifically for early identification of pathogen in cage culture or biofloc or anywhere. And we also EDN also shed in the light on the diets and trophic interaction in ecosystems. If eDNA can combine with the genetics, we can have the genetic diversity also, the natural samplers, so like sponges, or we can put some artificial collectors, so we can get the signals of the biodiversity. And the last point is aquaculture. As I said, there is a growing number of literatures and application in aquaculture and seafood industries. So in like every approach have some positive notes. eDNA have some challenges, and that we have to looking at and consider during implementation of eDNA in any region. So one of the major challenges barcode reference library. So we did the gap analysis of available species, fish species in the Indian waters, and out of that, we got the reference is from 45 to only 75%. Still, there is a gap. So these species from 75 to 100 or 50 to 100, we cannot get using eDNA because we don't have the reference. So these are the major challenges, and most of the tropical country, these problems still persist. The second thing is most of the environmental DNA works going on the temperate region. So we have to understand the ecology of eDNA. So eDNA is the molecule. So we have to understand this eDNA transfer from this place to this place, how it is transfer, because maybe it is false positive. We can get the eDNA from here and we can say, oh, okay, the X species found here, but it could not be from here. Maybe this eDNA flow transfer here. So we have to understand the ecology, then degradation, the source of eDNA, and also sedimentation. And if we are talking about the Indian fisheries or tropical water, there are many the pollution pollutants, so they can might be affect the degradation of this eDNA and also the temperature. So we have to understand the ecology. Then standardization method. Right now we don't have the universal methodology for eDNA analysis. So each region have to specify or to standardize the methodology based on their local regional scale. And the last one is the lack of supportive policy still is ongoing, because we are not seeing fish directly, we are capturing the indirectly the fish. So still there is a need of some supportive policy for implementation of this methodology. So for still is eDNA can be game changer for the Indian fisheries management? I must say yes, because it has some work in four principle only. So just the collect the water, then detect what you want, monitor on regular basis, and manage. That's the things. So finally, I conclude that eDNA, the environmental DNA, is not a one size fits all. It has many promising potential opportunities application, but still it has some limitation. It has own challenges that we have to look into consideration. So one of them is we discuss the reference lack of reference library. So it is a roadblock for implementing this analysis in any country, any region. Also the knowledge gap. So most of the work, as I said, in temperate water, so there is a knowledge gap to how this eDNA behave is from one species, and still we cannot identify the stages life cycle using eDNA. We just identify right now the species, which species are there. So these are the knowledge gap. So we propose right now in any in tropical country like India, integrating this analysis with existing methodology for monitoring and get the regional standardization. So these are the recommendation, and there is no doubt that environmental DNA will mark the shift and approach the Indian fisheries management, revolutionize it in any sectors, and it's a new future frontier for sustainable Indian fisheries management. And I'm thank you for this giving me this opportunity. Is my the team? Yeah, thank you.